Hello everyone, here we are, the team monitoring soil health in Lara's farm. We are testing most common soil properties such as soil pH, soil organic carbon, etc. But the key part of our experiment is to test soil biological conditions. And for doing this, we are measuring two things, the soil decomposition and the soil microbial diversity. The testing on soil decomposition will tell us how healthy are these soils that the microbes are quite successful in breaking down the organic materials. We're always expecting from healthy soil to decompose organic material to release nutrients and increase the content of organic matter, which will improve the conditions for agriculture and sustainable management. On top of that, the microbial diversity measurements will tell us what's the impact, for example, of land uses and management on these microbial communities. So we found two interesting options for measuring soil decomposition, either by following the experience in North America, digging White Andes, or following the experience in Europe, digging tea bags, which seems to have more scientific support. Tea bag index method involves the digging of two types of tea bags, rooibos tea and green tea. The tea bags should be kept in the soil for about 90 days, and over the differences between the initial and final tea bag weights, we can estimate the ability of the soil when processing the decomposition. Later, these results can also be analyzed together with other soil properties to understand, for example, what property help on making this process to be faster, or if we are storing more carbon in the soil as a product of the decomposition process. Since May 2019, we had run 2 consecutive incubations in the field, over 58 sampling sites in Laras, and we're still waiting for a third set that will be sampled next month. The sampling sites include all different land uses, soil types, and environments we can find across the farm. So far, we have seen 2 interesting results. The first one is the negative effect of droughts on decreasing the soil decomposition, and the second one is the great variability among different land uses when responding to this. Initial and final tea weights for measurements in August in 2019 and February in 2020. We can see the differences between both incubations. August values were taken after 3 dry months in Narrabri, and the ones from February were taken after incubations in wetter soil conditions. The lack of soil moisture has slowed down the decomposition, as well as increased the variability among the sampling sites. The second graph describes how the decomposition dynamic is different among different land uses: drylands, pastures, and forests. The axes show the decomposition rate and the stabilization factor S, which can be an indicator of the stabilized carbon in the soil. It's interesting to see how the relationship between both factors is quite specific for each type of land use, even despite the evident separation for drier and wetter soil conditions, according to our measurements in August and February. In this case, we can see how drylands have higher decomposition rates, but likely pastures are storing much more carbon. These results are consistent with our measurements of higher organic carbon in pastures in the beginning of the study. In the microbial diversity evaluation, forests show a significant high diversity of fungi and a low diversity of bacteria, which can be related to a lower pH in these environments. However, pastures and drylands didn't show significant variations. What's more interesting to see, however, is how the nature of these soil microbial communities is quite unique in each land use and soil type. These beta diversity maps represent in similar colors those microbial communities that are more similar to each other, while the most different ones are in contrasting colors. For example, contrasting communities of bacteria are seen between drylands and those forests in the southern side of the farm. We think that soil decomposition and microbial diversity are quite sensitive for monitoring soil conditions in Llara. These indicators would help us to understand what's a sustainable future for these soils, and we are looking forward to further results.